Okay, this is one of the component 3 mocks. No idea, completely lost track, don't know which one it is. So, it's question 5. It's the one that makes me want to cry. I hate this kind of question. Stupid plants with stupid bits of tissue removed. Hate it. Anyway, so you need to read the information which tells you what's going on. So we've got transport of sugars. Immediately I'm thinking phloem. Dr. Schofield's laughing at me because he loves this kind of thing.
No, he doesn't.
In the
No, I don't.
But he's a botanist. In the stems of actively growing wooden shoots, the leaves were removed from the shoots above the region to be investigated. Don't know why. And they're divided into 3 groups. So plant A has all the tissues outside the xylem. So phloem, everything, and we can see here we've got a little hole. But the xylem's intact, so it will still carry water. In plant B, the xylem was removed. That means we've got no water transport. At the base of the region, this left the phloem and the cortex, except for so we can still do sugar transport. And then they've helpfully not given you a picture of plant C, the control not shown. Each stem was enclosed in a clear glass cylinder. The cylinders were filled with distilled water. Well, why on earth you would do that? Which was changed daily. I really honestly am feeling I've gone through this paper before. Anyway, Dr. Sowell's denying it and making me do it again. Some cruel and unusual punishment. Cylinders are filled with distilled water, so you're filling up this with that, so that's got to be to do with so that they don't no water loss from the site. A bit like this principle of putting an impermeable thing over a burn. No sugars were detected in the water at any point, so the phloem doesn't leak. So we've got our lovely pictures, and then part A, suggest how plant C was set up as a control. So they've not given you a picture because they want you to describe it. So I'm thinking do not remove the vascular tissue. So vascular tissue should not be removed. And keep all other conditions the same. And I'm just going to give a couple of examples here. So for example, light intensity, I'm going to call it air movement rather than wind speed. Effectively want everything else that could possibly affect the transport to be the same. Oh, it's all on single sides. Cool. Right, I'm going to turn that one over like that.
Small consolation.
So, explain when the leaves were removed above the region being investigated. So, no leaves, I'm thinking no photosynthesis. So, there's no photosynthesis where leaves are removed. Can't even remember where they were removing them from. Removing them at the base of the region. Alright, so above. So no sugars are produced above region investigated. Okay, suggest why each stem was enclosed in a glass cylinder containing distilled water. I don't think of any other reason except for it stops it drying out. Stops the exposed cells drying out. And why change it daily so it doesn't get all bacteria-y, I imagine. So the water was changed daily to prevent bacterial contamination. Oh, it says it's continued. Do you know what? Let's have a look and see how long. Oh, it's only 5 minutes. Feels like a lifetime. Anyway, here we go. The results after running the experiment for 6 days. That's botany for you, shown in the table below. So we've got, I've forgotten what plant is doing what. I'm just going to flip back now. So, whoa, plant A has got its phloem removed. And this is a good plan actually. If you're in an exam and it's asking you about something that's 3 pages back, just transfer that information over. So there's the control, so that's what normally happens. You've got an increase in stem length of 68.2 and sort of medium amount of sugar in. Where you've taken the phloem off, it's much less, so that's your trend down there. And then your total sugar kind of goes up and down, but there's less in the phloem, much less, much less. So that's the least, and that's the most, and that's kind of normal. Okay. So, I'm going to just put the word no in front of phloem. Right, okay, explain the difference in results between A and B. So A and B are nice and simple. A has no phloem to carry any sugar. So above the region, again, just to remind you, by putting this back down. So this is where we've no leaves and with no phloem to translocate it. So it's not going to go anywhere. And then the xylem one, obviously you can still make sugar and you can still transport it because you've still got your phloem. So between A and B then, what can we say? Explain the difference. So phloem is involved in, let's use the right term, translocation of sugar. Removal of phloem prevents the transport of sugar past the cut. And I think, you know, again, using data, I would definitely perhaps put something in an exam about, you know, it is only 0.08 compared to 3.21. That will generate marks, usually. I'm just going to stick to the point here because I'm going to need to talk about growth as well, because there's 3 marks, one for presumably saying something about phloem, or it could be a data mark, and one for saying about the sugar and one for saying about the length. The stem grows less, and I'm going to put 8.3 millimetres compared to 68.2 in the control. And then I need to give a reason for that, so I'm going to talk about respiration. Because there are less sugars for respiration. Then of course I've just run off the end and they're going to cut that off. Right, between B and C then. So B, we're looking at no xylem compared to the control experiment. So we start off with what it does. So xylem transports water up the plant, or you could say from the roots to the leaves. removal of xylem reduces growth of the stem. You can see that on the table. But we need to explain why we've got more sugar. This means less sugar is needed for respiration to produce ATP for growth. And so the sugar above the cut region is more. So you're really explaining that because it's growing less, it doesn't need as much energy for growth, therefore there's more sugar that's not used in respiration. So that's why you've got more sugar in that one. Let's hope the next bit's a bit easier. With reference to the results table, suggest how the reliability of the results might have been improved. Repeat the experiment. Three times and calculate mean values. State two variables which should have been controlled. So this is the idea that anything, so I've given a couple of examples here for my control experiment. So it doesn't ask me to explain, it just asks me to say what they are. So I'm going to control light intensity. If I was asked to explain, I'd say that it would affect the rate of photosynthesis and therefore sugar production, or it would open the stomata and affect the transpiration rate. And I think also, you know, the one that we all love, temperature, please don't abbreviate that to temp. You could have had age of plants, size of the cut, humidity, air movement, any of the things that affect water transport or sugar production, leaf area, all of that kind of thing. Notice it's two things for 1 mark. Okay, oh, hurrah! At the end, look, ha! Diagram shows a cross-section of a root from one of the plants. Where would you find the Casparian strip? So, working from the outside, epidermis, cortex, endodermis. Endodermis is where I would find the Casparian strip. I need to make sure my arrowhead lands in the middle of that. Don't, you know, sort of do something over here. It needs to be in the middle of the structure labelling lines. And here we've got the apparatus for root pressure, so that leads on from the Casparian strip. So we've got a capillary tube, the stem, and it's pushing stuff up this ruler. Then cyanide, what do I know about that? No respiration, no ATP, no active transport. That's immediately what I think when I see cyanide. Root pressure falls. With reference to the role of the Casparian strip, so I need to say something about that's role in active transport, explain how we get a reduction of root pressure. So, the Casparian strip, what does it do normally? Forces water and minerals from the apoplast pathway into the symplast. Moving minerals across the endodermis requires ATP for active transport. This is pretty stock answer, isn't it? Cyanide stops ATP production. So less minerals, perhaps none. Can we cross? So the water potential gradient, I don't know why I started writing that, I should have just used the symbol. The water potential gradient is less, so root pressure is reduced. And you could have just said, so less water moves into the xylem. Hurrah! End of question. Woo. Can't find the book.